Thank you everyone for attending. My name again is Afreen Allam, founder and CEO of SiNoN Therapeutics. And today I'll be talking to you about increasing bioavailability by targeted drug delivery to treat neurological diseases. So as we all know, the real challenge that we face today is that only 2% of drugs and nanoparticles combined are able to cross through the blood-brain barrier. And naturally, we need things to keep stay out of our brain when we're healthy, but when a patient has a disease, whether it's brain tumor, Alzheimer's, even Parkinson's, we need medications to get across. Roughly 20 to 40% of tumors end up metastasizing into the brain, and every 67 seconds, someone develops Alzheimer's. Our patented solution is the carbon nanodot, where you can encapsulate small molecules as well as large biologics. And then using the Trojan horse mechanism, it bypasses the blood-brain barrier to deliver to a target site. But since it's carbon-based, the immune system is not reacting or having an adverse reaction to it. And once the nanoparticle has deloaded the drug or the therapeutic that's been encapsulated, it gets filtered out through the kidneys and gets excreted from the body. We've done some initial animal trials kind of looking at passage of the blood-brain barrier. So for example, this was done in a glioblastoma. Glioblastoma is one of the most aggressive forms of brain tumor today, with extremely low penetration, but also very low. There's typically been only one medication that's been used since the 1980s. And what this image shows is all the green cells are the tumor cells tagged with GFP, and the white line going down the center is essentially the blood vessel or even the blood-brain barrier, and all the red particles you see towards kind of distributed throughout the cell are the carbon nanodots. And what this trial specifically told us is that one, we're able to penetrate through the blood-brain barrier. The nanoparticles are actually self-fluorescent, so we didn't attach any receptors, we didn't use any pegylations or wrappers, and that's where you see all these red particles fluorescing near infrared. And we saw a pretty good uniform distribution throughout the brain. Next, when you're looking at competitive technologies in this field, there are many companies and many technologies kind of working to solve the same problem. And so what we did is kind of group them into classes or types of technologies rather than specific companies. So anything that has quantum dots or gold nanoparticles have a metal-based component, and eventually the pegylations will come off, leading to kidney toxicity and liver failure. Proteins and ligands are safer, relatively speaking. However, they're very specific to one molecule, one drug type. Nasal drug delivery is also a safe option. However, it's very limiting on the size of what you can actually deliver. And there's also the mucous membrane interactions, which you have to take into consideration before giving a therapy that way. Stem cell therapy is obviously the safest option available. However, with a patient that has just been diagnosed with brain tumor, we don't have the luxury of time to start from scratch for each individual patient to see how can we create a therapy that may or may not work for each patient. The poliovirus is actually being used as a delivery tool at the Duke Brain Tumor Center. And it's highly invasive because they drill a hole directly into the skull and inject into the brain. It's used as a last resort for many glio patients. However, as you can imagine, just by doing so, it opens up a whole new host of infections for patients, and recovery time becomes a lot longer. With our nanoparticle, we're using it as a platform drug delivery tool, so we're pretty much agnostic to which neurological disease we can focus on. Our first indication that we're building our proof of concept with is brain tumors. However, this can be applied to anything that needs to get across the blood-brain barrier. So far, we've seen things up to 500 kilodaltons we've been able to encapsulate. And as I mentioned, they are non-toxic and self-fluorescent, which adds to a dual capability of a target drug delivery tool as well as an imaging tool. So with the current market, in simplest forms, the easiest example we can kind of visualize is chemotherapy, where you're essentially pumping in a high dose of toxins, hoping the drug reaches the target site. In the long run, we end up doing a lot of damage to the healthy cells as well. And what we're focusing on at SiNoN is how do we bring down the overall dosage and the side effects, but improve the therapeutic effect of the drug itself. And the mission around our technology was focused around quality of life for patients. So we wanted to ensure We kept a patient healthy enough to kind of maintain normal activity and not be, strapped down to a hospital bed for the next month, which typically a lot of cancer patients go through this treatment cycle because they're so immunocompromised. And continuing on some of our safety trials, so for FDA clearance is a big concern with any nanoparticles. Once you inject it into an animal, ensuring that it's actually been able to be excreted was key. So we saw there was a peak around day 3, and by day 6, about 99% of the nanoparticle had been excreted. We looked at excretion samples, kind of been able to pick up the fluorescing capabilities, and that's the way we were monitoring that the nanoparticle was not degrading, but also being completely excreted from the body. On the toxicity side, typically a lot of our market competitors were given a dose of about 175 milligrams per kg, and that's dependent on the body weight of the mouse. And that's the dose we started at, kind of expecting to work our way down. Again, since we're carbon-based, the immune system was not having any sort of reactions. We had to keep increasing the dose until we got to 1,098. That's 5 times typical dose, and typically we would never give a patient or even an animal that kind of dosage, but at least we know it's not the nanoparticle that's causing the toxicity. And this also gives us a lot more flexibility to work on different payload optimizations for different therapies, and based on which indication we're targeting and which therapeutic we encapsulate, this toxicity level will fluctuate. But this is purely for the nano delivery tool by itself. We also wanted to look at, kind of looking at how can we increase the bioavailability. So for drugs, for example, paclitaxel, which has extremely low bioavailability, especially when it's injected or even in oral form, it started degrading within 25 to 30 minutes. And what we did is we encapsulated the Taxol within our carbon dot, which is the peak that you see over here. And we were able to extend the bioavailability, or even the concentration within the blood plasma, for about 4.5 hours, which significantly increased the time of when the circulation continues and before the drug itself actually starts getting metabolized. So this will be key for drugs that have extremely low bioavailability and efficacy is not strong enough, or you don't reach that threshold before you start seeing any improvements in the animals and eventually in humans. So this is kind of being applied to not just neurological diseases, but in general, focusing on the bioavailability piece of it. What we did is we were able to engineer these nanoparticles to have a slow release mechanism. So we didn't want to dump the entire payload as soon as you inject it into an animal. And in the case of a tumor model, it was very pH sensitive, so we can control the release mechanism based on that. Obviously, for something like Alzheimer's market, that's something we're still working on to figure out how can we potentially induce a release mechanism where it's not pH dependent, or there's not specifically a physiological change that we can detect. And just, the fact that there's really not enough known about the mechanisms of how Alzheimer's works, and there is no medication that we can currently test to even look at efficacy models. And so we were looking at the release mechanism, and so far we've been able to control it to 2 hours. This can be increased or decreased dependent on the target molecule, but as well as the target disease area that we're focusing on. Our business strategy is really to partner with pharmaceutical companies or even biotech companies that typically have things in their pipeline that are not passing through the blood-brain barrier, or you see extremely low passage. Versus medications that are failing in phase 2 of clinical trials because of the toxicity levels of how dependent the doses are so high. And so looking at both of these categories, how can we encapsulate it, make the drug more effectively delivered, but improve the efficacy of the drug? And it's really a 3-tiered approach where there's smaller developmental contracts, and then the real revenue stream for SiNoN kind of comes in with the milestones based on successful trials in the preclinical model and eventually clinical trials. And down the road, there will be royalty payments, but that's kind of, we're looking at another 3-4 years. The partnership process really looks like, we are a chemistry and engineering-based lab, so we focus heavily on the engineering aspect of the nanoparticles. We can work on new target molecules, and it takes about 3 to 4 weeks to turn around to give you something that we've been able to optimize the payload capacity. We will then also do the tunability to make sure that we get the right release mechanism based on the different indications we're working on. We can handle the in vitro trials, but what we develop or what we focus on our partnership is the in vivo trials and the preclinical models, since that's not what we heavily focus on. And we also will work with our future partners to work on scalability and producing them at a larger scale, but also making sure we're GMP compliant. We already have had validated interest from big pharma, kind of ranging from different fields like brain tumor and Alzheimer's, but as well as the reformulation side. So things that are about to come off patent within the next 4 to 5 years. How can we increase the patent life for something that's already been known to work, but also just improve the efficacy side or improve the delivery aspect of it. And we've also looked at, potentially using this as a delivery for large biologics, which has been key for some of the big pharma. And we're in diligence, but also completing some of our PK and PD trial data. We recently signed a contract with IIT, which is the Indian Institute of Technology, on the research facility. So we're working with them heavily on the characterization of the nanoparticles. So as I mentioned before, the nanoparticle that we produce is a platform. So currently our focus has been within CNS, so things that need to be used for target drug delivery, specifically being able to pass through the blood-brain barrier and using the controlled release mechanism. But we also have another capability of the imaging side. And through our initial trials, we've been able to show that the nanoparticle tends to aggregate towards tumor cells, or it has a better affinity kind of penetrating through the tumor cells. And what this tells us is that as tumors start to grow and spread, the nanoparticle will also be able to be detecting specifically early stage tumor cells, where things like if a patient has already had stomach cancer and now is already starting to have cells spreading to the pancreas or the liver, but they're not showing symptoms. Through scans, we can pick up on where the nanoparticle has, kind of spread to, and then look at the imaging capability. So potentially using this as a diagnostic tool. That's still in the early phases. For other therapeutic areas, we're looking at oncology as a whole. We've also been able to encapsulate insulin, potentially using it for oral insulin delivery. So kind of just in a high level overview, the blood-brain barrier delivery market's estimated to grow to about $4.6 billion by 2026, and 85% of those sales would just be in the US alone. We already have 2 patents. So the first one's been issued that covers more how to produce the nanoparticles themselves and what makes them water-soluble, self-fluorescent, and non-toxic. The second patent is more a method of use, so kind of looking at the encapsulation process, the release mechanism, and what different applications we can apply this to. This is currently pending, and we're hoping to hear back within the next 2 to 3 months. For research publications, we have 2 papers that were published in the Royal Chemical Society. And our current team has a background in biology, physics, and chemistry. We also have advisors from the Fuqua School of Business helping on the finance and strategy piece of it. We are looking to add on someone who has more business development as well as FDA expertise. So that's something that if you know of someone or if you'd be interested, please reach out to me and we'd love to talk. And of funding to date, most of the trials that we've done so far have been self-funded. We also won the Duke Startup Challenge while I was a student there and the NC IDEA grant. We've used this funding to complete our toxicity and clearance trials, but really focus on the payload optimization and increasing bioavailability. And we've been able to test the nanoparticle passage of the blood-brain barrier in 3 different animal models. The first was a glioblastoma, secondly, Alzheimer's, and third, just healthy mouse model. And this was to make sure that it's not because the blood-brain barrier is compromised, and that's what's allowing it to pass through. And our future steps are to raise a $2.5 million convertible note. These terms are still kind of being worked out, but just as a rough idea, this is what we're looking at. And this will go directly towards completing our efficacy trials, hiring business development and FDA consultant. We attend a lot of conferences to really increase our network with potential pharma partners. And as a biotech company, focusing on IP portfolio is always key. So thank you for your time, and I look forward to answering any questions.
Thank you, Afreen. That was very good. We do have some questions for you.
Sure.
The first one comes from Xinghong Yan. He says, we know in disease scenarios, the blood-brain barrier can be damaged. How's the penetration of your technology in healthy brains?
Yeah, so that was one of the last things I mentioned in the last slide. So we were looking at basically, there's not a way I can go back. Okay, so we were looking at, where is the nanoparticle tending to aggregate, and we saw they have affinity towards neurons, which eventually will be useful for Alzheimer's treatment. And we saw pretty much a uniform distribution even in the healthy cells versus the glioblastoma, which does have a blood-brain barrier that is compromised.
Okay. Next question comes from Greg Oakes. He asks, is the release mediated by the thickness and/or rate of degradation of the carbon dot? And how stable are they once manufactured?
Sure. So we can, they are multi-walled carbon dots. So we're able to control, not specifically, it's going to have 10 coatings versus 100, but we get a range of the size reference of the nanoparticle. We can produce them anywhere from 10 nanometers all the way to 250. We've seen the optimal absorption into the brain is around the 80 to 100 nanometer range. In terms of kind of the release mechanism, what we do is we engineer them by attaching functional groups to the surface, which are pretty stable, even kept at room temperature. The only time that we need to be temperature, or it is temperature sensitive, is when the molecule that we've encapsulated, starts degrading, and at that point, then we are basically running against the clock. And so far, we've seen the stability of the functional groups have been, pretty standardized or pretty kind of uniform throughout most of our trials.
Okay. A follow-on question from Xinghong is, what's the brain to plasma ratio in your delivery?
That is something that we are currently testing with the distribution and the PK trials, kind of looking at being able to quantify. That's something that we don't have facility to do in hand in our lab. So we're working with a facility that we third-party contract that we haven't got the results for yet, but we're hoping that we'll have them within the next couple of weeks. But things have been kind of slowed down because of COVID, and testing has been put on hold.
Certainly has indeed. You're right. That's the extent of the questions, Afreen. So thank you again very much for your presentation.